تعالوا نكمل ال third level وال fourth level of protein structure. احنا اخدنا ال primary structure وال secondary structure وال super secondary structure. Tertiary structure is the third level of the protein structure. For example, for the globular protein, it's defined as the steric relationship of the amino acid. Yeah, steric means space أو three-dimensional structure. The three-dimensional structure of any protein is called the tertiary structure, arrangement in space. The three-dimensional structure of the globular protein is dependent on its primary structure, الامينو اسيد اللي موجودة فيه. فهي لما تبقى arranged in space تعمل لنا it's connected by specific hydrophobic bonds. When the hydrophobic side chain تبقى tends to be interior in the structure and the hydrophilic group tend to be on the surface on the protein molecule. فاكرين لما قلنا hydrophilic و hydrophobic أو polar و non-polar؟ قلنا hydrophobic يعني don't like water, so it tends to be interior in the molecule away from the surface. Hydrophilic means like water and can form a bond with water molecule, so it tends to be on the surface of the structure. يبقى the tertiary structure of any protein depends on its content of the primary structure, in which the hydrophobic groups to be interior in the structure away from the surface, and the hydrophilic group tend to be next to the surface of the structure. Three-dimensional structure conformation of the globular protein, it provide and maintain the functional characteristic of the protein. Let's see the bonds that stabilize the tertiary structure. احنا قلنا each level of the protein structure have specific bonds stabilizing it. There are four types of bond stabilizing the tertiary structure. It includes the disulfide bond we mentioned before in the beta bond or the beta loop. Hydrophobic interactions, we just explained it. Hydrogen bonds, which is present in the secondary structure, and ionic interaction, it's a new bond, we will explain it. So there are four types of bonds that stabilize the tertiary structure. Again, disulfide bond, let's see in the figure. The disulfide bond is between two molecules of cysteine which contain sulfhydryl bond forming cysteine to form disulfide bond. And hydrophobic interactions, which include the hydrophobic non-polar amino acids like alanine and phenylalanine, tends to be interior in the structure of the protein in a three-dimensional arrangement of the protein, and the hydrophilic will be outside the structure. And there is the hydrogen bond. There are two types of hydrogen bonds formed in the tertiary structure. The hydrogen bond of the secondary structure between the carboxylic group of one peptide bond and the amide nitrogen of another peptide bond forming hydrogen bond. This is the dotted one, hydrogen bond. There is another hydrogen bond can be formed between the hydroxyl hydrogen of the serine. It's one of the hydroxy amino acid, do you remember? and the oxygen of the carboxylic group of one of the carboxylic or acidic amino acid like aspartate. So this is hydrogen bond. يعني ايه hydrogen bond؟ the hydrogen enter in it. It's between the hydrogen of the hydroxyl group of serine, one of the hydroxy amino acid, and carboxylic group of one of the acidic amino acids like aspartate. This form hydrogen bond. And at last, the ionic interactions. Ionic means there is exchange of ions, so it must be between the charged group. Do you remember the charged amino acids? It's the basic amino acids and acidic amino acids. Acidic amino acids have negative charge, and basic amino acids have positive charge, like lysine and aspartate will form the amino group having positive charge, and the carboxylic of the lysine and the carboxylic group having negative charge of the aspartate will form what's called ionic interaction or salt bridges or ionic bonds. This is the four type of the bonds in the tertiary structure. Let's see another figure. This is the hydrogen bond between the amide and nitrogen of one peptide bond and the carboxylic group of another peptide bond, forming hydrogen bonds. Even can be formed between the hydroxyl hydrogen of serine hydroxy amino acid and the carboxylic oxygen of the acidic amino acid like aspartate. It's hydrogen bond. This is the first bond. Second bond is disulfide bond, when two molecules of cysteine containing a sulfhydryl group SH, when we remove this both hydrogen with oxygen, this is reduction, forming water, and will form cysteine containing the disulfide bond. This is the disulfide bond after reduction. Next, hydrophobic regions that contain hydrophobic amino acids like alanine and phenylalanine tend to be interior in the structure of the protein, away from the surface. This is hydrophobic bonds or interaction. And at last, the salt bridges or ionic bonds between the charged basic amino acids containing NH3 positively charged and the charged acidic amino acid containing negative carboxylic group COOH, and this will form together ionic bond between charged amino acid, the basic and acidic. This is types of the bonds in the tertiary structure. In details, disulfide bond is a type of the covalent bond. It's strong bond stabilizing the protein structure, occur between two molecules of cysteine containing sulfhydryl group to form cysteine containing disulfide bond. It's covalent bond stabilizing the protein structure. Next bond, hydrogen bond, either between the carboxylic group of one peptide bond and the amide nitrogen of another peptide bond of the two different peptide bond forming hydrogen bond, or the hydrogen of the hydroxyl group of one of the hydroxy amino acid like serine, and the carboxylic group of one of the acidic amino acid like aspartate or glutamate, forming hydrogen bond also. The ionic bond or electrostatic bond or salt bridges It's formed between the oppositely charged side chain group of the polar amino acids, the basic, histidine, arginine, lysine, and the acidic, negatively charged amino acids, aspartic or glutamic acid, forming ionic bond. And the last type of interactions or bonds is the hydrophobic and hydrophilic interactions. Hydrophilic means water loving. They are associated with water, forming bonds with water, so they tend to be on the outer surface of the protein. Hydrophobic means doesn't like water, so tends to be interior in the protein, like hydrophobic nonpolar amino acids like alanine and phenylalanine. The last level of the protein structure is the quaternary structure of the globular protein. It's the special arrangement of the subunit of the oligomeric protein. It's linked by non-covalent bonds in the protein molecule. This forms the quaternary structure, like this figure of hemoglobin molecule. It's tetrameric protein. Tetra means formed of 4 subunits. Penta means 5 subunits. Hexa means 6 subunits, and so on. Di is 2, tri is 3. So hemoglobin is tetrameric protein formed of 4 subunits. These 4 subunits are connected to form the quaternary structure of hemoglobin, which is formed of the 4 subunits, and this is called the quaternary structure. The association of the subunits is called the quaternary structure, and not all proteins are polymeric. يعني ممكن بروتين يبقى just polypeptide chain, just alpha helix or beta pleated sheets, just tertiary structure forming motif or domain, مش كلهم forming quaternary structure. The function on dissociation of this subunit will be lost. يعني لو أنا جبت hemoglobin molecule عملت dissociation للفور subunits بتاعته, its function will be lost. The forces that stabilize the quaternary structure are برضو bonds, type of bonds stabilizing quaternary structure: hydrogen bonds, ionic or electrostatic bonds, and hydrophobic bonds. يبقى only 3: hydrogen bonds, ionic electrostatic bonds, and hydrophobic bonds.